Hello students, welcome to EPG Pathshala. I am Vidya from All India Institute of Medical Sciences. Now I am retired. My main introduction about myself is that I have been teaching biophysics for over 40 years and in various institutes like AIMS, JP Institute of Information Technology, DY Patil Institute of Biotechnology and Bioinformatics, and teaching is my passion. Today, we are going to talk on a module DNA conformational changes using bioinformatics tools. This belongs to the paper on bioinformatics. After completing this module, you should be able to introduce yourself various bioinformatics tools which are used in the conformational changes studies of DNA. Then we will like to discuss use of quantum chemical and molecular electrostatic potential techniques for studying of DNA. We would like to discuss some conformational studies on DNA. I would like to explain you the use of different energy minimization and molecular dynamics, that is EM and MD techniques for study of conformational changes, sugar repuckering, PZ transition in the DNA molecule. I would like to also enumerate use of MD or molecular dynamics technique for global structural changes in DNA. DNA molecule, as you all know, under different environmental conditions and also with the non-random sequence can adopt plethora of backbone conformations which are known as A, B, C, D, S, Z. Actually, practically every alphabet is being used for the definition of the DNA conformation. DNA molecule also has short range local conformational changes and long range global structural changes which depend on the environment and the base sequence. What does it do? This makes this molecule very suitable as a carrier of our genetic information, which is also amenable to the environmental control. DNA conformational changes can be monitored by a number of bioinformatics tools. We show in this picture 12 base pairs of DNA in different polymorphic structures. The first one in the extreme left is a B form of DNA, which is realized for random nucleotide sequence in a highly hydrated state. What happens if the hydration lessens? In lesser hydration, say less than 30%, DNA assumes a form that is shown in the middle. Alternate pyrimidine-purine sequences in high salt conditions adopt a left-handed or a Z structure that is shown in the extreme right. As you can see that these have different heights, their grooves also have different shapes, the sizes of the grooves are different, and many other physical differences between the polymorphic forms of DNA. What is really conformation energy optimization? The basic assumption in all conformational techniques is applicability of universal law of energy minimization imposed by classical Newtonian mechanics. Most interestingly, the origin of conformational methods lies in the celebrated work of Professor G. N. Ramachandran from Bangalore, who used hard sphere potential to obtain what is called Ramachandran map or a phi-psi map. Later, many people worked on this conformation energy minimization using atom-atom energy function and other people, scientists who modified these techniques. We show in this slide the potential energy function that can be used in general, most of the calculations. So total energy, potential energy, composed of various components: non-bonded, U non, electrostatic, U electrostatic, torsional, U tor, H bonding, bond stretching, bond angle, and improper angle deformation. These are composed of various terms. Like, for example, the non-bonded potential is typically known as Lennard-Jones 612 potential, is given as A by R to the power 12 minus B by R to the power 6. Whereas electrostatic potential is given as 332 Qi Qj, where Qi and Qj are the charges on the two atoms I and J, and Rj is the distance between them. A torsional potential depends on the torsional angle, barrier to the rotation, and the characteristics of the bond rather. So it has different forms. It is a generalized form is shown here. The hydrogen bonding potential given by Sharaga's group is also similar to non-bonded, but it is called 10-12 term. It has an attractive part and a repulsive part. Repulsive is positive. In both this non-bonded and H-bonded, Aij or Aaij or Bij and Bbij, these are constants which depend on number of other parameters. U bond, U angle, and U improper. It is the contribution because of the deformation in the case of bond length, bond angle, or out of plane deformation of some fixed torsional angles. Now, the parameters of these various terms can differ, and these terms are fully parameterized. This full function is used in energy minimization programs, molecular mechanics programs, molecular dynamics programs, Monte Carlo calculations, and the parametric form and the values is called the force field, and they have been given name after the name of the discoverer or some other thing. Like popular force fields are charm, amber, chromos, CVFF, CFF91, MM2, OPLS, etc. Details can be found elsewhere. Let me explain what is scanning of conformation energy space. In the conformation energy geometry optimization, first, the geometries are generated using internal coordinates r, theta, and phi. The energies are scanned not by changing all these parameters. We change only 2 or 3 torsional angles simultaneously. And then this data of energy with respect to 2 or 3 torsional angles is scanned and plotted as two-dimensional isoenergy contours like geographical contour maps. What is kept constant is the bond lengths and bond angles. That is, r and theta are kept constant and not changed. Now, can we go on allowing number of torsional angles to change? No. If the number of torsional angles is greater than 3 or 4 or maybe 6, then one has to use number of statistical methods to scan this conformational space. What we need is a starting geometry, because the energy has to be dependent on the geometry. Then only we can use this methodology to obtain the geometry of the molecule. So one has to start from some geometry which is based on some information. Like generally we use chemical bond lengths, bond angles and dihedral angles. And this can be done by using structural data on fragments or any closely related molecule. One can also use crude structural data or explore structural databases as Cambridge Crystallographic Data Center CCDC at HTTP CCDC that came AC UK, or Protein Data Bank, conventionally used as rcsb.org, Nucleic Acid Structure Database at Rutgers. These are all available at the public domain and you can download these structures freely. Conformation energy calculations, one use not the complete atom-atom potential described earlier, but generally 2, 3 or 4 terms in the potential energy function. For small molecules or molecular fragments, one can as well calculate energy using quantum chemical methods based on molecular orbital calculation or MO methods, and there are a number of such methods: CLO, CNDO, INDO, MINDO, MINDO3, AM1, IEHD, EHD, PPP, UCAL, Pi, UCAL Pi, Del Rey Sigma, etc. There is an advantage of using MO methods because once you set up computation of charges or computation of energy based on the MO methods, one can also do some other computation which is very, very important for DNA. Like we can calculate accurately the charges and the atom centers from the wave functions or molecular orbital wave functions. We can also study electronic aspect of nucleic acid structures. We can plot electrostatic potential or MEP maps in 3 dimension or 2 dimension. Molecular mechanics, one can in principle use either internal or Cartesian coordinate system. Internal coordinate approach is very cumbersome, but it is closer to the chemical structure of the molecule. So sometimes it is preferable. A typical example of such method is a damp least square algorithm or DLS. This technique was developed in our group only. Starting from Wittek's method, we developed a DLS method. Cartesian coordinate optimization techniques are more popular. They use several different minimization methods or minimizers. Steepest descent, SD. Newton-Raphson minimizer or NRAP. Conjugate gradient, CG. Variable matrix quasi-Newtonian minimizer or Powell, POWE minimizer. Molecular mechanics packages generally offer you option for minimizer as well as different force fields, so you don't have to be worried about it. In molecular dynamics, the trajectories or dynamic behavior of a molecule is studied under the influence of a force and kinetic energies. Unlike energy minimization, here we look at the trajectories of atom under the influence of forces exerted on them by other atoms and their kinetic energies. There are several theoretical procedures that highlight different aspects of molecular dynamic behavior. But the most commonly used by all people is a simple molecular dynamics. It's generally called only MD. In simple molecular dynamics, the trajectories are generated on the basis of position and velocity at a particular time, considering the molecular system to be Newtonian and conservative. In simple terms, it means that acceleration is equal to force upon mass, and force is equal to the first derivative of the potential energy. And what we try to do is calculate the temperature, and if the temperature changes, like temperature is calculated from kinetic energy, and kinetic energy is equal to half mv square. So if the temperature changes, you can rescale the velocity. And total energy E, which is a kinetic energy T and a potential energy, is a constant of motion. In MD simulation, we integrate equation of motion and compute trajectories for each individual atom as a function of time. So we know how the system is behaving together. We need to simulate the system at constant temperature. We can also simulate at constant pressure. But for this, we have to rescale velocities or couple the system weakly to a temperature path at a temperature theta 0. Individual derivatives and integrals are calculated trivially with respect to Cartesian coordinates. Various algorithms, Verlet, Gear, Leapfrog, etc., are used for integration. Molecular geometry optimization methods belong to also some other classes. One very famous is a Monte Carlo simulation method by Metropolis. This method generates the configuration of the system by making random changes, not systematic changes. Randomly they pick up and they make random changes in the position of the species present together with their orientation and conformation. There is another technique, simulated molecular dynamics, also known as SAMD. This method explores various states by generating small random changes in the current state and then accepting or rejecting each new state. This had been very successfully used in case of gases and liquids. There is another very interesting algorithm, and this is based on our biological systems. This is called genetic algorithm. In a genetic algorithm, or GA, there is a global optimization done. And in that optimization, we always accept or reject. The successful or good conformation or good things are accepted, that is survival of the fittest. Fit conformations are accepted and some are rejected. This can be compared with the simplex or stochastic methods and is very popular. The quantum mechanical parameters such as delocalization energy dE can be calculated on the basis of energies of highest occupied molecular orbital and lowest unoccupied molecular orbital. For example, dE for adenine, guanine, cytosine, uracil and thymine was calculated to be equal to 3.89, 3.84, 2.28 and so on. dE for AT base pair was found to be 6.11, whereas for GC it was 6.33. The sum of isolated constituents was 5.94 and 6.12. This gives extra stability of 0.17 for AT and 0.21 for GC base pairs, which is in the same direction as the melting temperatures of AT and GC base pairs, which are 70 degrees and 110 degrees. Molecular electrostatic potential, or MEP, deals with the potential around the complex molecule, and it is very, very effective because the electrostatic potential around these molecules is crucial for their interaction. Again, we can use isopotential contours like geographical contour maps, or there are methods to plot the whole surfaces by choosing different colors, and we can find the site of electrophilic attack for a particular molecule, which is very helpful to design a new ligand. We know that this ligand can bind at this particular site. The specific activity of the drug depends on the compatibility of the two potential surfaces, not only structure but the electrostatic potential surface compatibility. Molecular electrostatic potential map are calculated on the basis of Coulomb interaction between a unit positive charge placed in the vicinity of the molecule and that on the atom centers in the molecule, obtained by any molecular orbital method, and it at any point xyz, it is given by the equation uxyz equal to minus qi upon rij, but sum over all the atom center. The technique becomes complicated if we use ab initio and other molecular orbital methods, but it gets better because it gives better result. The reason is, in molecular orbital, the charges are not centered on the atom center, but it is a cloud or distribution is there, and the MO vectors are used for calculation of this function, potential energy function U. It is also possible to compute first derivative of the potential energy and find the direction of the electric field, MFA, which is very useful. MEP, or molecular electrostatic potential map, of a isolated GU Wobble pair and its comparison with the Watson-Crick GC or AU base pairs was studied by Zhu. He used amber charges and used Poisson equation. The electrostatic pattern varies due to change in the backbone conformation and the base geometry. We show in this figure MEP, the molecular electrostatic potential map, for GC base pair, a Wobble GU base pair, and an AU base pair. In these pictures, the yellow color shows the most negative, minus 1.7 kT by E. Green shows the most positive, 1.7 kT, and red intermediate negative, while blue intermediate positive. If you look carefully in these three views, along major groove, axial view, and a minor view, you can notice definite differences between the GU base pair, GC base pair, and AU base pair. GU has enhanced negativity compared to GC or AU base pair. This may be important for predicting binding of cationic ligands to RNA molecule. Jairam's group in IIT Delhi was one who studied MEP map around B-DNA in great details rather. The detailed description of DNA charges, DNA charge distribution, as we know, polarizabilities of the macromolecule and the solvent was used using Poisson-Boltzmann distribution. And he used the method which is known as finite difference method. DNA surface was mapped on the surface of a cylinder. The DNA is cylindrical. What the map was created not as a circle or a plane, but it was created on a cylindrical surface. The cylinder was then cut along the axis and then opened, and then the maps are presented. Solvent in this case was treated as a continuum of dielectric constant 80, and surrounding region was mapped in a mesh of 65 into 65 into 65 mesh points. We show in this picture molecular electrostatic potential map around B-DNA in 3 different sequences. First is poly-DA-DT, second is poly-DG-DC, and third is GC-GAA-TT-CGC. In this picture, the weakly negative regions, which is the potential greater than minus 0.5 kT, are cross-hatched. Strong negative regions, which are less than minus 3 kT, are shaded. One can notice differences in the group potential among 3 DNA sequences. Detailed shape of DNA as well as the existence of dielectric boundaries have been found to be important to affect the group potential in the case of DNA. You can see clearly that the central top picture and the bottom picture, they show difference in the group potential. We show in this picture the backbone sugar phosphate for nucleic acid structure. As you can see, it consists of all single bonds, not like protein, and that's why it is highly flexible. So it has torsional angles which are flexible, and these are classified as sin means minus 180 plus and minus 90, anti means 0 plus and minus 90, G plus or Gauche plus 60 plus and minus 30, G minus or Gauche minus minus 60 plus and minus 30. We also show in the picture the IUPAC nomenclature for the backbone torsional angles. If you start from phosphorus P, PO5 dash is alpha, O5 dash C5 dash beta, C5 dash C4 dash rotation is gamma, C4 dash C3 dash, it is restricted, but it is called delta, and C2 dash O3 dash epsilon and O3 dash P as the zeta. So alpha, beta, gamma, delta, epsilon, zeta. In the case of sugar rings, these are, for the 4 rotations are described as nu0, nu1, nu2, nu3, nu4. We show in this slide the backbone torsional angles IUPAC notation. Shown here are the 4 atom sequences which define each backbone torsional angle, alpha, beta, gamma, delta, epsilon, zeta. Their values for B-DNA and A-DNA are given. We also show torsional angles in the sugar ring and their values for B-DNA and A-DNA. Just let me tell you that the older terminology for the glycosyl torsional angle, chi-CN, was little different. So you'll find in some papers use that. So there, the glycosyl torsional angle was defined as O4 dash C1 dash N9 C8 in purines, and in pyrimidines O4 dash C1 dash N1 and C6. Their values for B and A DNA were 84 and 26 respectively. There are many theoretical studies which have been reported using classical empirical potential energy functions and also quantum mechanical methods as EHT, CNDO, CELO, ab initio, etc. and backbone conformation of nucleotides and DNA, dinucleotide, trinucleotide were studied. Different authors use different models. Like some people used only dinucleotide monophosphate, some people use dinucleotide triphosphate, etc. The results are usually presented using two-dimensional contour maps for rotation around two adjacent backbone bonds simultaneously. We show in this slide conformation energy map for rotations around PO5' and PO3'. Again, the older nomenclature, people called these angles as omega, omega prime, whereas current nomenclature we use alpha or zeta. The maps look different because the potential energy function, the methodology is different, and the models are also different. For example, in Olson and Flores map, they used empirical potential and used dinucleoside monophosphate model. Whereas in Saran and Goville's map in 1963, they used mononucleoside and also empirical potential. And the energy minima you find, which was at 180, 180 in the case of Flores map, shifted to G minus G minus or G plus G plus, where G minus G minus is in the agreement with the experimental points 290 degrees to 90 degrees. We show here some more conformation energy map for the same rotations, omega, omega dash. But you find again they are much different than what we saw in the last slide. Now, in the case of Srinivasan's study, 1973, the lone pair interaction of nitrogen was considered in diribonucleoside monophosphate. And MAP has shown one important thing. Although it looks similar to Flores map, the trans-trans TT region, which was low energy in the case of Flores map, is no more present in the case of Srinivasan's map. Whereas in the right-hand side figure, the Goville and his group, they changed the potential energy function and modified it. And the equation is shown here rather. They used diribose triphosphate model, but then the results, not only they are very close to experiment, they also agree with ab initio calculations and CELO calculations by other workers. The glycosyl bond conformation was studied in Pullman's group using CELO technique for C3-endo and C2-endo sugar puckers with ribose nucleotides in the absence of hydrogen bonding. It showed that these nucleosides have preference for anti-conformation with chi-CN from 0 to 90 degrees and for C3-endo sugar pucker, which get restricted to 40 to 70 degrees for C2-endo sugar pucker. The purine nucleotides had greater probability to be in anti-conformation than syn conformation, which agreed with the experimental observation. Coleman group used energy minimization method to examine structure and energies of local conformational changes in DNA. We show here typical helix axis, long axis, and the dyad axis about which the rotations were monitored. The relationship between local twist angle and backbone conformation was examined by them. They also examined DNA melting curve. Number of authors studied short-term molecular dynamics, that is less than 100 picoseconds. MD simulation of nucleic acids in vacuum showed structural degradation. That was a very difficult point. The structural stability could be improved by scaling all electrostatic charges to 0, which is not physical, but it improved the results. Scaling dielectric constant to mimic explicit solvent and counter ion condensation. This is needed because DNA is never present as naked. It is always in the presence of solvent, or by inclusion of explicit hydrogen bonds and water molecules. There were also studies using explicit solvent and counterion condensation. There was also some other approach using periodic longitudinal boundary condition, and this effectively was equivalent to considering a segment of infinite long double helix. The backbone torsional angle variation in the molecular dynamics simulation of 100 picosecond is shown in this picture in the forms of conformational dials. Here the center of this circle represents 0 picosecond, while the periphery represents 100 picoseconds, and angles are plotted. And you see that some angles show more variation than other angles. We find also base pair tilting, helix axis movement, H bonds movement, bifurcated H bonds, and H bonds between different base pairs were also seen in such kind of dynamic studies. Short-term molecular dynamics was studied by number of authors, which are shown in this table, and the motions of individual atoms, that is their amplitudes, anisotropic in the motion, unharmonicities was monitored, hydrogen bond dynamics was monitored, dynamics of the backbone torsional angles was monitored. It showed variation from 6 to 12 degrees, 9 to 21 degrees, and 14 to 23 degrees for different torsional angles. There were several structural changes noted. One is spontaneous repuckering of the sugar ring. Sugar puckers, we know that C2- endo, C3- endo, over 4- and exo, and like that rather. So McCommon and Harvey, they showed that spontaneous repuckering needed only few picoseconds, and the average amplitude was about 0.04 nanometer. The pathway between B and Z DNA transition was studied by Olson's group and Harvey. The vibrational entropy difference between the B and Z form of DNA conformation was about 91 joules per mole inverse. It was of the same order as observed by experiment. People studied global structural changes in DNA. For example, Levitt studied using MD optimization of number of base pairs needed for straight DNA and bent DNA. And he found that straight DNA, the optimum solution you get for 10.5 base pairs per turn, and for bent DNA, which is normal condition in the nucleosome, you need about 10 base pairs per turn. Overall time bending model was also studied by Ellison and McCommon. He modeled the chain as a string of bits with full treatment of hydrodynamic interaction. Bending stiffness of DNA was reported with persistent length of 60 nanometer. The chain was stimulated for a very long period, 3 to 200 nanoseconds. Authors generated fluorescent polarization decay curve and model decay curves for structure factors from polarized dynamic light scattering. The model decay curve matched with the fluorescent depolarization predicted by theory, while light scattering experiments were found to be insensitive to the internal motions in DNA. So students, what we have learned in this module? We have enumerated bioinformatics tools used for the study of nucleic acid structures. We had discussed quantum chemical methods used for study of electronic aspect of DNA structure and molecular electrostatic potential maps around DNA. We have enumerated conformational studies on DNA, described EM or energy minimization studies on DNA, described use of MD simulation for local structural changes of DNA, spontaneous repurposing of DNA, sugar puckers, BZ transition in DNA. We also discuss use of MD simulation for study of global structural changes in DNA. I thank you very much.